Another important analytical technique, which is basically a chemical analytical technique and is frequently used in molecular biology, is the mass spectrophotometric ionization. That is the mass spectrophotometric ionization technique. The principle is that ions, they may be produced from a neutral molecule by removing an electron. When an electron is removed from a neutral molecule, what happens? That it is converted into an ion. And it produces a positively charged cation because the electron has been removed from it and now it has become a positively charged cation. And or we can do another way by adding an electron to form an anion. That is, we can add an ion or electron to produce an anion and remove an electron to produce a cation. Now, when it happens, then there is an electron impact ionization, that is EI, and it is widely used for the analysis of metabolites, pollutants from the environment, and pharmaceutical compounds. So when we carry out this technique, we can study the metabolites which are produced, we can study the different pollutants which are present in the environment, and we can also analyze the pharmaceutical compounds for different purposes. For example, in drug testing programs, electron impact, it has major application as a mass detector for the gas chromatography. That is, for the material which we use for the gas chromatography to identify this electron impact, it has the major application for mass detector there. So what happened? A stream of electrons from a heated metal filament is accelerated to produce 70 electron volt potential. In this case, electron impact to generate the electron impact. Then the sample ionization occurs when the electron stream, it is across a high vacuum chamber produced and they are allowed to diffuse. So the heated metal, it produces those ionization, and then in a highly vacuum chamber, they are allowed to diffuse there. Just as shown in this figure, there is a filament, electron beam, sample is produced on the top, and in this chamber, the ions are there, and we get plus 70 electron volt in the vacuum, and then the ion is focused. There are ion focusing by strong magnets around, and the mass analysis detector, which detects what is the different molecule, the different weight of the different molecule. So in nutshell, electron impact source, it is depicted in this diagram, that how it is carried out in the mass spectroscopy. Now, there is an interaction with the analyte, or analyte is the material which we are going to analyze, and it results in either loss of an electron from the substance, or it captures an electron, or vice versa during the process. Now, this method is also applicable to the peptide as well. We can study different peptides which are present, and before the advent of electrospray or the MALDI, which we have studied earlier, the method did have some applicability to the peptide. It means that, as a matter of fact, we have to study the proteins which are important biomolecules and their different mass of the different peptides which are present, and this technique can be applied to that. Now, this can also be used to study the chemical bonds. The nature of chemical bond of organic molecules, they are formed by pairing of electrons, and then the mass spectroscopy can help to determine the chemical bond, its nature, which is present among those biomolecules there. Now, ionization resulting in a cation requires loss of an electron, as we have already discussed, that whenever there is a loss of an electron, what happens? A cation is produced. And from one of these bonds, it leaves a bond with a single unpaired electron. Where there are pairs of electrons, that is the ionization. With the ionization, it leaves with the bond with a single unpaired electron, that is, there is a electron which is not pairing, and it leaves from there. Now, the radical ions, they are termed as the molecular ion parent ions. Molecular ions are the parent ion precursor ions. So, they can be termed as molecular ions, parent ions, or precursor ions. That is, the three types of the radical ions can be made or can be present during this process. Now, chemical ionization is used for a range of sample similar to EI, that is the electron impact, which we have studied earlier. That is the chemical ionization, and EI, it is also produces the range of samples which are similar to those. Chemical ionization is useful for the determination of molecular masses. That is, when we have to determine the mass of the molecule, the chemical ionization can be used and will be used there. Chemical ionization contains suitable reagent gas such as methane or ammonia that is initially ionized by electron impact, and the high gas pressure in the source results in ion molecular reaction between reagent gas ions, some of which react with the analyte and produce the ions of the cation. That is in contrast to we saw where we have provided a high vacuum and high voltage. We can also have a high gas pressure which can do the same, or which can do the same source results as a ion molecular reaction between reagent gas ion to produce the analyte or to produce the results.